Hello, and welcome to this Genomics England Innovation Showcase. In this session, we're going to be talking about the genomics of COVID-19, both the virus and the genomics of the human genome, to help understand how some of the work we've been doing at Genomics England together with people across the UK life sciences sector have helped us to understand some of the puzzles around COVID-19. I'm Rich Scott, I'm the Chief Medical Officer at Genomics England, and I'm joined with me in this session by two people. Firstly, by Professor Sharon Peacock, who is a Professor of Public Health and of Microbiology at the University of Cambridge, and in her role at Public Health England has established the COVID-19 Genomics consortium, COG UK, which has been the driving force behind establishing the UK's potential to sequence the COVID virus itself. I'm also joined by Dr. Kenny Bailey, who is a research fellow and a consultant in anaesthesia and in critical care at the Roslin Institute and at the Royal Infirmary in Edinburgh. And as I say, we're going to talk about the work that we at Genomics England have been doing with Kenny, with Sharon, with many others across the UK life sciences sector, looking at COVID-19 and the genomic determinants of disease. We're going to be talking about that from two perspectives. Firstly, the genomics of the virus itself. So the COVID-19 virus has an RNA genome. That's the nucleic acid code that its genome is written in, and it's about 30,000 DNA or RNA bases long. Sequencing that, as we know, has helped us understand the biology of COVID. It's been really important in designing vaccines, and also, as we've all become intimately aware over the last 18 months, it's helped to understand where new variants of the disease have emerged and to track them. We're also going to be talking about the sequencing of the human genome and how that has helped us to understand why some people get severe COVID and some people get more mild disease. So reading, in that case, the 3 billion letters of the human DNA code in people who've had COVID. And in the case of the work that we've been doing here in the UK, comparing the genomes of as many as 20,000 people with severe COVID in hospital, in intensive care with COVID-19, with 15,000 people who've had COVID more mildly, and comparing those and understanding what drives those differences in terms of those participants' genomes. And that can help understand the biology. As you'll hear from Kenny, it's also been useful in making real decisions about working out which medicines might work better to prevent severe disease. So we're going to be hearing about that. We're also, I think, going to be hearing about how crucial collaboration across UK life sciences sector has been in this, and particularly at the beginning of the pandemic, how fast decisions and collaboration really set us on the front foot in the UK in terms of genomics. I think we'll also hear about the importance of the historical strengths of the UK in terms of genomics in putting us on that front foot. And that's not just the early discoveries that have been made in the UK in terms of genomics from Franklin, Watson and Crick, creation of new sequencing technologies, but it's also more recent investments and ongoing investment that keep us at the head of the game in terms of genomics worldwide. So, for example, us at Genomics England, having benefited recently from the sorts of investment that allowed us to build the infrastructure to deliver the 100,000 Genomes project, also to have that infrastructure to support the NHS Genomic Medicine Service whole genome sequencing service, and how we can use that infrastructure that was there and invested in to quickly adapt it to respond to the COVID pandemic. We'll also, I think, reflect on what the lessons we've learned are and what that means in terms of making sure we're prepared for future pandemics and remain ahead of the game in terms of genomics in this pandemic. So what we thought we would start with is going to Sharon. Sharon, I wonder if you'd like to introduce yourself and then perhaps start by reflecting on and telling the audience some of the stories about the beginnings of the genomics response to COVID-19.
It's absolutely wonderful to be here, so thank you very much for the invitation, and I'm really pleased to be able to talk about SARS-CoV-2 sequencing in the United Kingdom, and actually more widely, because it has been so important for our pandemic response. And so I'm a professor of public health and microbiology at the University of Cambridge, and I also direct the COVID-19 Genomics UK consortium, which is conveniently kind of abbreviated to COG UK. And to date, that consortium has actually sequenced more than 1 million viral genomes, which is really quite an unprecedented number of genomes, which has helped to inform decisions, as you mentioned. But where did it all start? Well, it really started with an early conversation with Sir Patrick Vallance, who's our government chief science advisor, and a very wise man. And we were saying that, really genomics was going to be key to understanding the evolution of the virus. Evolution is something that happens quite naturally. And we knew that after the virus emerged, that it would start to change. And we needed to be able to capture those changes so that we could understand the virus and its biological characteristics. Was it going to be more transmissible, or was it going to be more lethal, for example? So we needed that understanding. And so I've got a very nice image here of the initial, let me see if I can share my slides. So the early development, you can see a sort of pictorial representation here by a fantastic artist who in the bottom left-hand corner, following conversation with Patrick Vallance. Can you see those okay? Yep. You see the slides? Yeah, okay. You can see in the bottom left-hand corner that we called together mid-March 2020, actually, when there were fewer than 80 diagnosed cases in the country. We called together a group of scientists from across the United Kingdom who were experts in pathogen sequencing. And 20 people met in a room at the Wellcome building in Euston Road and actually discussed how we would put together a sequencing network. This was unprecedented times. We pulled together everyone who we thought could contribute to this. So there we are in the bottom corner. We put together over the course of a day a blueprint. So often when scientists get together, they'll talk about the nitty-gritty of science, but we talked about how could we practically put together a sequencing network for the United Kingdom. So within a few days, by the 18th of March, we had developed a blueprint, which we passed to Patrick Vallance. And we were very fortunate to get supported by government funding through the COVID-19 Fighting Fund. And, the rest, as they say, is history. You can see in the image that we connected with the Lighthouse labs and other testing labs to get samples for sequencing. And we did the sequencing, which goes into a database, and then we use the genomes to understand how the virus is evolving. So this is what we developed. So if you look at the map, hopefully there's a sequencing facility near you, and you'll see that we have 16 regional sequencing sites across the UK, and they're in actually universities and the 4 public health agencies of the United Kingdom. And the Wellcome Sanger Institute were key for this too. They're a major sequencing site, and the project was administered in Cambridge. Now, you've mentioned that this is really important. Why do we need so many genomes? Well, actually, going back to the very first genome, in February 2020, this was a genome that caused an infection in a patient, or a virus caused an infection in a patient in China, and was released on the 3rd of February. And that was actually really instrumental for the very first vaccine design. So from that sequence data, you can tell a lot about the virus. You can tell what sort of virus it is, and it's kind of what sort of proteins it produces. And it was really important for vaccine development right from the very beginning. And we've already mentioned the fact that going ahead, this is really important for monitoring for vaccines, and that is very much the case into the future for as long as the virus is circulating. So I've got other slides that I can show in terms of how we've tracked the virus, but I think what I'll do is I'll stop there and perhaps come back to my slides about how we tracked the virus a little bit later.
Thank you, Sharon. I'm really interested by to know a little bit about sort of maybe what some of the history was that helped you when you entered that room and had those discussions and came up with that blueprint. What were some of the things that either had been done before or had been thought about before that meant you were able to make such good and rapid decisions?
Yeah. Well, I think you have to go back to 2013, actually, when there was an announcement about the 100,000 Genome project by David Cameron. And that really raised awareness and started to build the infrastructure for sequencing. And actually, that was focusing on a combination of rare diseases and cancer. But a third work stream was actually pathogen genomics. I think at the time it was less well talked about. But that was focusing on pathogen genomics, and I actually chaired that group. And we made a set of recommendations about how we should start to use sequencing technologies for pathogens. And so we made recommendations that tuberculosis, the cause of tuberculosis should be routinely sequenced. It gives you really rapid information about resistance and also outbreaks. We also indicated that HIV sequencing, deep sequencing, that's having really detailed information about emerging resistance to drugs, going to be important. And also sequencing of hepatitis C also for treatment. So, right back in 2013, we were putting in place mechanisms to sequence pathogens. And I think following on from that, there was an awareness in certainly politicians' minds, policymakers, and that kind of drive also developed our capabilities. I think key moments were when Dame Sally Davis, our previous Chief Medical Officer, wrote her annual report, volume 2, and in 2 of those, she had one on genomics, which was really important for us in pathogens because we could highlight what we did. But we also got involved in the antibiotic resistance volume 2. And I think since 2013, and even before that, actually, we've been building our capabilities in the country so that by the time the pandemic came along, we had a really good group of experts across the country. We had the equipment, we had the expertise. We had to refine our methods for SARS-CoV-2, but it was like we were sort of in the starting blocks by the time SARS-CoV-2 came along, and were ready because of that history actually to get started with sequencing the virus. And by the time we got funding from the government, we'd already sequenced something like 250 genomes.
Fascinating, thank you. And I guess coming to you, Kenny, perhaps you sort of to introduce yourself and give a little bit of background both to the work that that sort of preceded COVID-19 from your point of view in terms of understanding determinants of people needing critical care for various reasons, and how again that set you and us as a country in a really strong position to respond rapidly to COVID-19 when it did arrive.
Well, thanks, Richard. So I agree completely with Sharon. I think that the key message, the key learning point from this outbreak is that what worked was what was already working. So we built on the foundations of existing activity, and without activity, infrastructure deteriorates. For the host genome sequencing, we already had a project operational in about half of the intensive care units in the UK, and it was ramping up. We started it in 2015, and the objective was to use host genetics to understand the complex biological problems that we face in intensive care medicine, including sepsis, flu, and emerging infections like SARS. So we were ready. Of course, when the outbreak hit, as with in every other sector, we had to adapt quickly and scale up but the infrastructure, the protocols, the approvals, and the people were already there and active. And that's really why we were able to scale up so much more quickly than any other country. So many other countries put resource into host genetics during COVID, but none have created as much genetic discovery power as the UK. In fact, the UK study that we've done in collaboration with Genomics England has more genetic discovery power than every other study of the host genome in the world put together. And that's fundamentally because we were able to get the right patients in very large numbers. I do have a few slides that I can show you about those results, if you like, Richard, and then go back to the discussion. So I can show you perhaps what we found. I think that would be of most interest. We were faced with the problem at the very start of the outbreak that we didn't know how to treat COVID. Before it was even called COVID, we'd had extensive discussions of what treatments might be useful, and I had the privilege of contributing to the setup of the recovery trial, which I think most in the audience will have heard of, led by Peter Horby and Martin Landry at the University of Oxford. When we first started choosing treatments to put into recovery, we had very little evidence for which treatments to use. We knew that the host immune system, your own immune system, is likely to be damaging the lungs. We now know that to be true, but we weren't sure that that was the case. We weren't sure that immunosuppressive drugs would help people. And an even bigger problem is that the immune system is incredibly complicated, and we don't know which buttons in it to push, which targets to aim for with drugs. And that's really where host genetics, I think, has most to offer. As most people will know, 6 of the drugs that we've tried so far in recovery have not been shown to confer a benefit on patients. And by a combination of good judgment and good fortune, 3 of them have, and together these have more than halved the mortality in critical COVID. 2 of the treatments that are currently being randomized right now, that we're currently testing in recovery, have been added to recovery, in part at least because of genetic evidence that we've provided from the Genomic study in collaboration with Genomics England. And I'll show you the results just very briefly in 2 slides of Genomics. So this is the result we got within 5 months of the first case hitting the UK. We had recruited 2,200 critically ill people, and that's important. The most extremely sick people with COVID, we recruited into the study and compared to the rest of the population. And this, I think I probably don't need to describe a Manhattan plot for this audience, but just for anyone who's not familiar with these plots, this is every chromosome in the genome lined up along the x-axis, and there are 5 million spots on this graph. The y-axis shows how sure we are that a given variant, a given genetic difference is really different between critically ill COVID patients and the rest of the population. And where it crosses this red line is where we agree that it's probably real. 4 of these, so you can see there are multiple hits and multiple points where it crosses the red line. We also set ourselves a second challenge, which is that we have to replicate them in a second group of patients. And 4 of these associations were robustly replicated, and we're very confident that they are real. And all 4 of them lead to therapeutic hypotheses, to ideas for treatment. Probably the most directly related to treatment is this gene type-2, which is one of the targets of baricitinib, a drug currently being randomized in the recovery trial. And actually, since we produced this evidence, that several clinical trials have been reported suggesting a benefit. And perhaps just as a teaser, this is what we found most recently. So that first slide had 2,200 patients included, and we'd used microarray genotyping, a cheap but fast technology to read the genomes. This is the results from genome sequencing in 7,400 people. So genome sequencing done by Illumina under the guidance of Genomics England. And this really shows the power of firstly, getting large numbers of the right patients into the study, and secondly, the resolution that you get from genome sequencing. There are 25 spikes crossing the red line on this graph, and all of them are indicating important parts of the biology of the disease. this paper is under review right now, but it's in the public domain. People can see it on medRxiv. I'll stop there, and perhaps we can speak about some of the other aspects. I've got a few other slides I can show if needed.
Thank you, Kenny. So I think I was really struck at the beginning of the programme about how integrated the thinking was, in the same way that Sharon talks about some of the work that had gone together, and how those people together planned out a blueprint for the genomic response, particularly in the virus. The close working of recovery of your study, GenOMICC, of other studies, for example, ISRIC4C, the study you lead, looking particularly at clinical data of people with COVID. We've also been working with a number of others across the country. So, for example, in the work we do with you, Kenny, we're also working with the REACT programme, so led by Paul Elliott at Imperial College, who are the team who people will have heard on the news, just as the ONS also do. They send teams to households across the country periodically, so that we can keep a track of the prevalence, the frequency of COVID in the population. We've worked with them to approach their participants, to ask them if they wanted to join the same programme, and so that we can then, alongside that network of recruitment that your team had already established ahead of the pandemic across intensive care units, across the NHS, and with so much input from the NHS itself, also recruit participants to the programme from home who have had more mild disease, and in some cases long COVID, so that we can both compare their genomes, severe and mild, but also begin to understand potentially more about the biology of long COVID.
Yeah, I think the collegiate interactions between different studies has been one of the defining features of the UK response to the outbreak, and really why the UK has led the world in both discovery science and therapeutics in COVID. The other studies that we absolutely depended on were UK Biobank, and I've just realised that my little video is over the UK Biobank logo on our slides, but of course they provided the control group for our first genetic study, and that was already there. That's infrastructure that already existed and was generously shared. And of course the 100,000 Genomes project, which Genomics England created, which provided the controls for the data that we have so far. For those 25 extra hits, we, I think, can look forward to much more discovery. We now have 15,000 patients in the genomic study, and with REACT and Paul Elliott and the Genomics England team have recruited 15,000 individuals who are well matched to the critically ill people, both in age and sex and genetic ancestry, and had relatively mild or asymptomatic disease. So I think the discovery, the power of the study will more than double in the next few months when we analyse that data.
I wonder, it'd be interesting, in fact, later in this collection of showcase sessions we have today, we have a focus on diversity of data sets. You mentioned there, Kenny, the importance of the diversity of the COVID participants, and being able to make sure that those discoveries are relevant to everyone. I wonder whether you can sort of, again, give us a bit of a flavour of some of the challenges, but also the work that you have been doing with the team in the more recent work with the whole genomes.
Yeah, thanks. So I'll show this slide again because it contains a few hits that are exclusively discovered because we had both whole genome sequence data and a more diverse population than in the first study. Now, in genetics, everything, the discovery power is determined primarily by the number of patients, the number of the right patients, with the right clinical characteristics that you get into a study. And it's difficult to draw comparisons between a control group from one genetic ancestry and by that I mean a genetic heritage and a case group from a different genetic ancestry. So we're bound to compare the groups of people that we have the most of. And in the first study, that was white Europeans. Now, over the last 12 months, since we reported the results from those first 2,000 cases, we have several hundred patients from several other genetic ancestry groups that are common in the UK, and through the 100,000 Genomes project and the direct recruitment through the Genomics England programme and REACT, we have people to compare them to. And that means that we're able to see signals, for example, if I zoom in here in chromosomes 1 and 2, TRIM46 and BCL11A, these are genetic signals near genes that have these names, probably mediated by these genes, that have been revealed because we're able to compare between ancestry groups. And particularly here, you can see that this is all one spike. It's difficult to tease apart genetic signals that are next door to each other on the genome. And the reason is that a whole block of genetic variants are inherited together, but those inheritance patterns are different in different ancestry groups. So having different ancestry groups helps us to tease apart signals that are right next door to each other in the genome. And of course, the other thing that you need to do that is really high resolution data on the genome, so the whole genome sequence has made that possible. So that's, it's extra discovery that we couldn't really get any other way.
Thank you. And I guess it goes without saying, but crucial for both programs has been rapid funding decisions and sort of enthusiastic funding from multiple sources. So, for example, for the program we've been doing in human genomics of COVID, funding from the Department of Health and Social Care, from UKRI, which predated for the study and then supplemented that with specifics funding for the study, from NIHR, from LifeArc, and also contributions from Illumina have been crucial for us, for example, not just to fund the sequencing of the genomes, but also from a Genomics England perspective, one of the important things that we were able to do was establish a new cutting edge cloud-based research environment that meant that the data were not just generated, but generated and made available with the right tools to support these rapid discoveries, so that the genome data were generated, the consent process that participants went through meant that they were agreeing to join our National Genomics Research Library. So their genomes sit alongside the genomes of the 100,000 Genomes participants, and participants or people who agree to a research consent offer through the Genomic Medicine Service and other research studies, so their data can be made in a de-identified way available in a trusted research environment that approved researchers can then visit to make these discoveries. And it's that support, and not just the infrastructure to be able to do the sequencing, but also to have the right tech and tools available so that we can get the right minds and algorithms there to make those discoveries. Sharon, I wonder whether it would be a nice time to go back to you to talk about the discoveries and the work in the viral sequencing program.
Certainly, yes. So perhaps I'll just share my screen again and show a couple of slides just as illustrations. So, what has sequencing done for us? I think at the very top of the list, we need sequencing for, I've already mentioned vaccines and therapeutics, but just to sort of dig into that just a little bit in a bit more detail. It's kind of, when you're looking at viral evolution, then it's survival of the fittest, and you have a fit virus emerging that can outcompete other viruses. It's a sort of simple philosophy that the fittest virus would be the one that can transmit the most, and or the one that can get around our immune system, dodge our immune system. And so looking at the vaccines that we have at the moment, their original development was guided by the first genome that was released. And at the moment, they're highly effective, actually. But looking at future viral evolution, what we need to be clear about is that our vaccines remain effective in the longer term. So it could be that we need to tweak our vaccines to make them as effective as they are today. And already vaccine developers have developed vaccines against specific what's called variants of concern. There are 4 circulating in the world, and these include variants that kind of partially dodge the immune system. But the other thing that we need to do is to watch for the virus evolving resistance to other therapeutics. Now, Kenny's already mentioned some therapeutics, but what's called monoclonal antibodies, these are antibodies that have been raised in laboratories, but that can help in fight infection. They're given in cocktails, but actually, and to try to help people who haven't got a strong immune system to fight against the virus and against the infection. But actually, what the virus will do over time is actually evolve to try and escape the effects of those antibodies. And so I think top of my list for sequencing now and in the future will be keeping vaccines and other therapies working effectively. And that includes actually antiviral drugs. So that's really important. Now, the second area that sequencing is really important for is it really does help us to understand the wider characteristics of the virus. I've already mentioned managing to dodge our immune system, which could lead to a risk of reinfection or infection after you've been vaccinated. But actually, if we can group the viruses into variants of concern, then what we can do is then study those, work with our biology colleagues to study them to understand whether they are more transmissible, for example. And so we detected the alpha variant first emerged in Kent, and we know that that's more transmissible than the original virus. And the second virus that's been problematic in the UK is Delta variant, which is more transmissible than the alpha variant. And so we can understand transmissibility, but also we can look very carefully at the variant versus disease severity. So we know that 2 variants of concern increase the risk of going to hospital, but not the risk of dying from that infection. We also need to check that our diagnostics are going to keep working. So as the virus genetic code changes, that can sometimes change the way our diagnostics work. Now, it's been fortunate that actually none of our diagnostics have been rendered not effective anymore, but there have been some areas where the diagnostics perform differently, and actually we've used that to good effect, particularly with the alpha variant. We were able to track the alpha variant through the PCR result. So we also use these to track variants coming into the UK and spreading in the UK. So what this wonderful graph shows is really the pandemic since the very beginning, from March 2020 right the way through up to the end of July. And each colour represents one particular version of the virus, or we call variant or lineage. So every colour is a unique lineage. And it's normalised to 100%. So at any given point, it's a proportion of what's causing infection in people in the United Kingdom at that time. Now, if you look at the kind of white blocks, what you'll see is I've put on there superimposed wave 1, wave 2, and wave 3. So what we can see here is that in wave 1, there was a huge diversity in the virus. There were a large number of lineages, and not one was kind of particularly dominating. This was before the variants of concern emerged. Then we go into wave 2, and we can see there's actually at half of wave 2, we've got lots of diversity. We have that red blob in the middle, a lineage called B1177, and that increase in the UK, it kind of got up to about 50% of all of our viruses are causing disease in people here, was most likely due to European holidays, travel. So it first emerged in Spain, and this lineage spread around Europe. Now, fortunately, it didn't have any special characteristics other than it was traveling in people who were traveling. And then you can see the alpha variant emerge, and you can see how it becomes completely dominant in the country for actually a period of months. And it's only kind of pushed off at the top spot by the emergence of Delta variant, which was introduced by travellers and then rapidly spread around the country. Now, this is really important for planning. we need to know on a population level what variant is circulating, so we can then match our expectations for whether disease may be more severe or whether it's more transmissible, and there may have to be a titration of measures to try and stop the transmission, which we know is all to do with hands, the face mask, space between people, and kind of being outdoors, but also vaccination. So this is how we can track the variant, and I think that this image is kind of really elegant description of the variants that have circulated, which have a direct impact on decisions around policy, etc. I think that that's my last slide, so I will stop there and hand back.
Thank you, Sharon. And I guess one of the things we've got a feeling for through talking about the human genomic work is some of the interactions with some of the other studies that are going on. I wonder if you can tell us a bit about how that data is being used and accessed, sort of both by the sort of core teams, but also how it's interacting with some of the other data sets that are available.
Yeah, certainly. Well, I would start by saying that as a community, there's been an immense support for data being released immediately. So all of our viral genomes are released into GISAID, which is a global database. So what we have, we share immediately. But you're right, they have used the viral data in numerous different studies. And so we've been able to, for example, sequence the virus in vaccine trials to see if people get infected with SARS-CoV-2 after they've been vaccinated, what variant is that? You know, is there kind of an emergence of a variant that's actually escaping that vaccine? We've worked with people who have been investigating, for example, outbreaks in prisons and schools and in hospitals. We've also sequenced for the ONS study, which you've already mentioned, and REACT, so that we can tell at any given moment what is circulating in the country. And we've provided sequencing for studies in care homes and so on. So we have shared our data extensively. We share it with biologists because they need to understand how a given variant actually behaves, you know, in laboratory experiments. And we share this with immunologists. Anybody who wants this data can have access to it. But in looking to the future, I think sharing of these data sets with data sets such as the human genome, and I'm sure we're going to talk more about this, actually what that's going to do is going to lead to really novel analyses where, you know, there hasn't ever been, to my knowledge at least, you know, 1 million pathogen genomes combined with a very large number of human genomes. And the possibility for discovery and discovery moving into predictive medicine, I think, is immensely powerful. So we're going to we've done an initial analysis of the virus, what variants are there, what impact does that have on control measures and policy decisions and vaccine development. But I think we're moving into a new era now of where bringing these data sets together means we can actually go into discovery science and really understand the interactions between the virus and the host in much greater detail than we've done for any other disease.
And for us, one of the really key things about the consent, what the participants are agreeing to in the human genome study is that ability to link data, because that adds, as you say, so much power to what can be understood. And for example, our teams working with the COG UK teams, working through just the practicalities of that linkage to make it work, so that we can, as you say, combine viral genome data sets and alongside those human data sets. And often it struck me through this, it's the unsexy stuff of making multiple studies work together and link well, and just the conversations that actually add so much value, so that what's delivered from the broader UK life sciences sector on this is greater than the sum of its parts, and it's that multiplicative power through the linkage. I wonder, I know, Sharon, you've been quite struck by the human side of that in the interactions, and I wonder whether that's something that you could expand on.
Yeah, well, actually the initial interactions with Genomics England happened from day one, and so Sir Mark Caulfield was at our brainstorming event actually back in March 2020, and ever since that time, COG UK have worked really closely with Genomics England, with ISARIC and others, and we've built those teams, and those teams have been there from the very beginning. And so working together, I think you're absolutely right. It's the kind of the practicalities of how do we link data? Where is it going to be housed? What's the process for accessing that data? And it's that kind of really underpins our discovery science, which is going to be so important in the future. But actually that is also underpinned with regular interactions, good relations between people. And I think that the idea that we would bring viral and human genomes together, at the very beginning, March 2020, means that we've got plenty of time to plan and develop those kind of connections between us. So I think that this is kind of outside of my usual experience, actually, and I think it's a credit to scientists in the UK that they've been so keen to work across disciplines rather than stay in their own disciplines. And I think that's going to make a big difference for discovery science in the UK.
And Kenny, I wonder whether, I know you personally, but more broadly, the human genomics work has linked in with multiple other studies looking at different facets of the biology of COVID response. I wonder whether you can sort of share some of what's been going on and your reflections.
Yeah, I mean, I agree completely. What has really driven the innovation in the UK during COVID has been the boring but important stuff that we have been paying attention to for some time. So you mentioned the ISARIC study that builds on a consensus protocol for studying new diseases that we developed back in 2013 in the wake of the swine flu pandemic. The ISARIC consortium was formed, initially led by Jeremy Farrar, the head of the Wellcome Trust now. I chaired a working group that was tasked with coming up with an agreement on the data and sampling strategies that we should take for any new outbreak. And in fact, that was endorsed by WHO and established in multiple countries around the world. And that protocol was used for the very first description of the clinical features of COVID in January 2020 in Wuhan. In the UK, we established the protocol together with Callum Semple in 253 hospitals across the whole country, and it was there ready as a sleeping protocol, initially supported by Sally Davis when she was CMO, as the first urgent public health study. So when the first case arrived in the UK, we were able to recruit them into the ISARIC study, and since then we've recruited many patients. So we've obtained samples from thousands and data from 239,000 patients now. That data set is of great value for both public health, government planning and research, and has led to many papers that I think many in the audience might be familiar with. But I think the real power of it is the ability to link to other studies, exactly as you're saying. So we've already linked the overlapping data from the COG UK viral sequencing study to provide additional prospectively collected clinical data about the hospitalised patients for whom we have viral sequence data. And of course, the host genome data, we're now linking to viral sequence data. I think the boring but important element of that is that it is the human stuff that makes that work. It's we have extraordinary computational facilities. The UKRI funded outbreak data analysis platform here in Edinburgh sits at the UK National Supercomputer Facility, where there's about £100 million worth of hardware. But really what makes it work is humans linking the data sets fighting through the mismatching numbers, the missing data components, and the quirks that there are in the data, and building a data resource that is both comprehensive, which it already is, and usable, and that's an ongoing program of work supported by HDR UK. And actually, if there are people in the audience who are interested in using that data set, then they can apply for support from UKRI, HDR UK, and the Turing Institute through an open call right now. The data set comprises now 750,000 viral genomes, 240,000 clinical data sets, the genetic data from thousands of critically ill humans, and a range of other studies that have been generously contributed to the platform. That's a resource that's going to continue to become more useful as more people use it, because it's using it and working on it that makes it more accessible.
And on the theme of people power, one of the things that I've been really struck by and enjoyed during this process is talking to our participants in the National Genomics Research Library, who are at the moment predominantly people who joined through the 100,000 Genomes programme. We have, as I think you both know, a participant panel who are involved in our governance across Genomics England, including oversight of who has access to the data, but also the other decisions we make. And one of the things they've been both really eager to hear about, but also delighted and excited by, is the fact that, and you mentioned this, Kenny, is that their genomes that they agreed to be made available in this National Genomics Research Library could be powering some of these early discoveries. So they're people who joined our programme for other reasons. They may have had themselves a rare disease or a family member with, or they had cancer, but they want their genomes, they want their data made available in this secure way to drive exactly this. And it's that people power and the collaboration with others. You mentioned Biobank, etc. And it's the humans driving the really positive collaboration that we've seen, but also the people who are participants in these programs powering and actually really pushing us to make sure that the data's available and used to make really important insights.
Yeah, I think the interaction with patients particularly has really driven this work. One of the bits of advice that I got quite early in my career was from Steve Webb, an intensivist in Australia, who said, do the research that benefits your patients most, and all the papers and grants will follow from that. And there's nothing like being funded by patients to really force you to do that. And actually, I mean, you listed financially the largest contributors to the Genomic project when you were talking about funders earlier. And of course, they've made it possible. But the reason Genomic existed at all is because a small charity called the Fiona Elizabeth Agnew Trust sets its research, which is run by bereaved relatives of intensive care patients, gave us £120,000 to establish the study over 3 years. And they were very flexible about how it was used, and they didn't require outputs within the term of those 3 years. We told them it might take more than a decade to have impact. And it was that flexible funding that just trusting the investigators to take a risk and establish something that might take a long time to yield, that really made it possible to deliver Genomic. And of course, the source of the funding being from relatives of people we'd cared for in critical care really focuses everyone's mind on delivering something with real clinical impact.
And that actually moves really nicely to the sort of last area that I thought it'd be nice to talk about before we move on to the Q&A, which is about what some of the lessons are for the future. Sharon, I think you talked a little bit about some of the sort of historical legacies we've had that put us in a really strong position. tell us, yes, share some of your thoughts on what this tells us for the future, and so we can make sure that we can respond rapidly to future challenges like this.
Yeah, thank you. Just before I do that, just going back to your previous point about the importance of people, I would say that something that we're very aware of in COG UK is that 1 million genomes is a huge scientific achievement, but each of those genomes actually relate to a patient who's had COVID-19. And we're always very cognizant of the fact that we need to think about the people who've been affected by that. So we have very much the same philosophy that Kenny's talked about, the importance of doing the right research for people, and we're grateful to have access to those genomes. Lessons learnt. Well, if there is one thing that I would say I would change about the pandemic if I could, and what I hope we will do well next time, is when a kind of unusual infectious disease emerges that could be the next pandemic. The lesson for me is that we need to study that very intensively. So we need to have mechanisms in place for clinicians to recognize that there's a kind of respiratory infection in somebody, but you don't know the cause. Could this be the next pandemic? And have the ability to sample that person to really understand what the cause is. And sequencing is only one part of that, but it's an important part because you need to understand very rapidly whether there's a new virus on the block that's causing an outbreak. Once you get that information, you can close that down really quickly and it doesn't become a global pandemic. So the very biggest lesson learned for me is that having the capability to diagnose people with sort of unusual or undiagnosed diseases is very important. And Kenny and myself and others have talked quite extensively about how you'd have that framework. Now, Kenny's framework does exactly that, but we need to actually have the ability to, as I say, sequence the pathogen. And if you know what it is, you can contain it very quickly. I think other lessons learned, the importance of team science has been really important. So scientists will often work in their own areas of speciality. I think working across disciplines, human genetics, viral genetics, studying the virus itself, virology, studying the immunology, that has really changed the way we think about disease. So I think a lesson learned for me is never just stick to your own tribe, but also look outside and look at what other people are thinking and saying. I think there are other areas. I think in terms of scale, I don't think that we will ever want to go back to very small scale sequencing because we've seen how useful it is. And so a lesson learned for me is we can use pathogen sequencing to great effect. And we need to now be nimble in thinking about how we're going to apply that going forward in relation to areas such as antimicrobial resistance control or the next pandemic. I think finally, I would say that I have become so much more aware of the importance of data linkage. And so as a pathogen genomicist or geneticist, I don't think that previously I appreciated the importance of data linkage so that you can link your genomes with your patient and with other data sets. And so for me, making sure that looking ahead, that the country is in the best possible position it can be in preparation for the next pandemic in terms of moving data across the country from patients into important data sets and then linking those together to make rapid kind of discoveries or impactful and actionable information that can help us contain and control the diseases. And I could talk. I could probably talk for a day actually on lessons learned, because I have to say that you have to have a sense of humble optimism as a scientist. I think humility, because you can't predict what's going to happen next. For example, I can't predict what's going to happen with the virus next. But optimism, because actually the science that we've developed, including the vaccines and the treatments, have been quite extraordinary in terms of their pace and scale. So I'm hopefully a humble optimist as we look forward, and that's also a lesson learned for me, that I can't know everything. That's okay, but I learn every day.
Thank you. Kenny, I wondered, sort of similar, your reflections on what the lessons are for the future and preparedness for the unknown.
Well, I agree completely with everything Sharon said, and I can't say any of that better than she did. So I'll only add that I think a lesson for me has been that there is a colossal amount of potential resource available in the academic sector. And when things really got difficult at the end of March 2020, the entire biomedical research sector in the UK certainly was able to refocus on a new problem, and that brought tremendous intellectual and practical resources to a problem, and it is one reason why we've made so much progress. I mean, we now know more about COVID than we do about any of the other diseases that I treat year in, year out in intensive care. So that adaptability is key, and what we already knew is that the outbreak you're preparing for is never the one that hits. So we were preparing for flu, and we got MERS, and we were busy with MERS, and then Ebola came along, and then Zika was the next big threat that came along. Completely different patient group and set of challenges, and then the one that we were all in the back of everyone's head since 2002 hit us in 2019. So I think adaptability is absolutely critical. Designing incentive structures for scientists to work together is a challenge that many people are engaging with, and that does need attention and leadership as it's getting from funders and publishers. The other thing that I think is maybe fits with my biases and I think should be a big lesson from this outbreak is the need to engage with clinicians. So Sharon mentioned that we need to empower clinicians to study what's wrong with their patients when they have unusual diseases. I think that has been a failing of research in many parts of the world, less so in the UK, but creating capacity for clinicians to engage meaningfully with scientists and study the diseases they're faced with, I think is deterministic for success. So critical care is the canary in the coal mine. It's the first place where all of the outbreaks that I listed before were detected. Unusual groups of patients presenting to intensive care, it's I think through clinical insight that really we've been able to deliver so much in the host genetic program by focusing on patients with the right clinical characteristics to optimize genetic power. We're able to make more discoveries with 7,000 patients than other groups have made with 30,000 patients in other studies around the world.
And I guess one of the reflections I have on the discussion we've been having is actually how new a lot of the technology we've been using here is. What's really nice about the way we talk about it is we take it for granted that we have these next generation sequencing technologies that we wouldn't have dreamt of having up our sleeves had the pandemic had happened even a decade earlier. We would have been in a very different place. And I guess recognizing both that the investment to mean that we have the infrastructure there to respond rapidly, to sequence 1 million pathogen genomes, to sequence at a drop of a hat tens of thousands of people's genomes who have COVID, it has been absolutely crucial. And also that science, thankfully, is moving fast, and being able to use the very latest rapidly is really important. So that sort of remaining at the forefront in terms of genomics and other aspects of science and technology that are relevant to the pandemic also sort of feels like something that we in the UK are fortunate to, I think, sometimes take for granted, and that ongoing investment really feels crucial.
Yeah, I think that's absolutely right. That's such an important point. We do take it for granted, but if I look at the cost and the pace of sequencing, if we go back to 2001, 20 years ago, it would take 18 months and £500,000 to sequence one E. coli genome. That's a bacterium that lives in our gut. And now you can sequence, for a very low cost, very rapidly. So you can sequence a genome in under a day, and perhaps £100, or actually often less than that, depending on how many you sequence at the same time. And so we have to be, we have to be mindful of the fact that has been a revolution in our lifetime. But what really excites me looking forward is if we've managed to achieve that much in 10 or 20 years, what's to come? And so I'm really excited by the prospect of new technology that allows us to do new things faster. I hope that the trajectory of discoveries and improvement is as rapid as it has been in the last 10 years, actually. I'd just like to come back to one point, though, which I'll be annoyed with myself if I don't make. Going back to lessons learned, one of the things that has been really obvious to me is that public health agencies and academia, universities working together has really made a big difference. We need to try and conserve that model, because no public health agency can have all the resources it needs in times of absolute crisis like we've seen in the last year and a half. Universities and academics can make a really big contribution to that. And for example, the Wellcome Sanger Institute has done a large amount of sequencing. So I think another lesson learned is really being flexible. Sorry to squeeze that in, but I was sitting here thinking, I really must make that point, because I think for our future preparedness, that model is going to be key.
Thank you. And a great one to end on. We've now got some time to answer some of the questions that have come in from the audience. So we've got sort of 15 or so minutes for the Q&A session before we close. So perhaps to start with a couple of questions on sort of preparedness and the UK response. But here, Courtney Jones is asking the same question, but really from a sort of a global perspective. What is it globally that we need? And perhaps linked to that, Sue Martin asks about, just for a feel for what international cooperation has been like, what it's felt like. I don't know whether perhaps we'll start with you, Sharon.
Okay, thank you. Courtney, looking at the global viral sequencing, what I would say is that it's actually critical for us to have viral sequencing across the world, because actually in areas, particularly in areas where they have low access to vaccines and high disease rates, this is the sort of environment where variants of concern could emerge, because there's plenty of opportunity to infect people when the virus actually makes a mistake in its genome and develops mutations. It's only when it's replicating in people. And so I'm particularly concerned about the emergence of a new variant of concern in areas where we have limited vaccination and no sequencing. So it's vital that not only do we vaccinate the world, but we actually have sequencing across the world. Sequencing is actually quite patchy in some countries. I can see that for example, across Africa, there's a big initiative, people working together. There was a recent fantastic paper in Science where 112 different research groups came together to describe SARS-CoV-2 in Africa. But there really isn't enough, so we have to do more. And in COG UK, we have a training program called COG Train, where we're providing online courses to help people to be able to sequence, together with train the trainer courses and online classrooms. That's not going to solve the whole problem, but we're starting with kind of education. I think for Sue, in terms of international cooperation, there has been very good cooperation. And one of the key areas really has been that people have released their genomes, their viral genomes, into a global database for everybody to see and everybody to share. And so from that perspective, there has been very good cooperation. And there's been some strong leadership from WHO in terms of genomics. They've been very helpful in terms of coming up with a new naming scheme so that we have names for variants of concerns rather than the places from which they originated, which was actually very stigmatizing. And so overall, there has been good international cooperation. However, there's a lot more to do. So what we'll need to do, when we went into the pandemic, we didn't have an international structure for global pandemic preparedness, and that includes sequencing. And that is what we need to do. So we've done quite a lot in the times of emergency, but looking ahead, we need something that's much more sort of robust and structured. And you may well know that there are discussions ongoing at WHO and elsewhere to develop pandemic radars and hubs for pandemic preparedness.
And Kenny, I wonder, you alluded to some of the international collaboration and in fact, in the context of the power of the UK work for host for human genomes here. Give us a bit of a flavour of what's been going on.
Yeah, so I mean, I didn't mention this so far, but GenomiC was always conceived as a global study, and we, from the very outset, took an open source model. So all of the study documents, the grant applications, the kind of source code that takes so much effort to write to make a study was completely open. And the intention was that intensive care doctors and clinical staff around the world would be able to pick that up and establish their own studies. Because we recognise that in order to tackle the really big problems in critical care medicine, we need to recruit over a very large area, and we need to recruit across the full spectrum of diversity of humans. So we are recruiting now in the UK, Ireland, Canada, and Brazil, and there are studies and set up across many other countries, including Australia, Pakistan, and Sierra Leone. I think the beautiful thing about genetics, about host genetics, is that the statistics force everyone to work together. You always find more if you combine your data. And so the genetics community have shared their data very openly, and been able to do international meta-analyses very quickly and efficiently. And really, there's the genomic study's been by far the largest of the contributors to the international meta-analyses, and there's been a very respectful and collegiate approach to that. I think in future, the founding principles of the genomic study that we state at the beginning of every collaboration are respect for sovereignty and capacity building, and I think they're the principles that we need to build on in order to engage respectfully with clinical and genetic researchers all over the world. Where we're building new capacity, it has to deliver something useful for the clinicians, scientists, and the population. So the approach that we're taking is to engage with an extraordinary group of people who lead an intensive care audit network, where they go to a new country where research infrastructure and critical care is completely absent, and establish at the beginning clinical audit, where all that happens is data recorded for each critically ill patient, and then that enables local intensive care units to compare themselves to their peers, see how well they're doing, and just the fact of doing that drives up the quality of care for patients. But of course, it also creates the infrastructure where that we can then build on for just getting a consent form signed on a single sample, which is fairly straightforward for a genetic study like GenomiC. And then, of course, building on to doing randomized clinical trials. So building that infrastructure is possible all over the world. That's already been demonstrated. And what it needs is investment and momentum.
Brilliant. So I'm hoping we might have a chance for just a couple more questions. So one that actually I'll take is a question from Mohalb, who is interested about how we ensure subjects' genomic privacy is maintained in the context of us talking about their whole genome, so the person's whole genome. So the approach we've taken in this study is the same as the approach we take generally across the work we do, where participants are engaged in a choice to join our research programs, and on the principle of people understanding what we do when we hold their data, it being their data. And what we do with the whole genome data is we de-identify it, so we take away sort of direct identifiers. Of course, we still need to be very careful because a whole genome is a whole genome. And the model that we use is one which we often refer to as being like a reading library approach. So we have a trusted research environment where the data is held in that de-identified fashion. Accredited researchers who declare what the research they are planning to do are able to access that data, to visit that data, and only take away the metadata, the summary data that they've derived, the results they've derived, and so that maintains that control over what is very personal data. I just wonder, Kenny, there's one more question that I think people would be interested in about the relevance of this work. You mentioned that it built on previous work, including on Ebola and other emerging infections. How will the work you've done here help us respond rapidly to sort of other infections as they emerge, like that, or Ebola and so on specifically?
I hope that it will. I mean, it's generated momentum. So we now have particularly data infrastructure in the UK for linking the data sets that we've talked about, but also we've demonstrated that this stuff works. So we've demonstrated that genomics can suggest host treatments. We already knew from other diseases that more than doubles the chance a treatment will be effective. Sharon and the COG UK team have demonstrated the extraordinary power of of pathogen sequencing. So I think we've shown what works. In terms of infrastructure, we've spoken a lot about the UK. I think it's very unlikely that the next outbreak will arise in the UK. So there's a tremendous amount of work yet to do. Just as it's never the disease you expect, it never arises in the place where you expect it. So building both surveillance and operational research capacity around the world is going to be a major challenge, and we'll need continued leadership from WHO and an open and adaptable mindset from clinicians and scientists.
Brilliant, thank you. So we are just sort of coming to time, so I want to thank you, Sharon, thank you, Kenny, for joining us. I've really enjoyed the conversation and sort of been interested in where it takes us. I think the themes of collaboration across the community, I think that crucial theme of people power, not just the community, but of the people whose data, who've agreed to have their data included to power these findings, which as we've seen have, and we've heard in the news day to day how important these discoveries have been. I think it's so striking what has happened in this pandemic, and that humble optimism you talk of, Sharon, I think is a really nice and important sentiment to take away. I've also been struck, as we've talked, by the importance not just of the wise historic investment in infrastructure, but also the importance of ongoing investment in infrastructure, whether that's keeping us at the forefront of the technology, but some of those other aspects that we've talked about, so that we remain in the UK both a sort of a real hub for UK life sciences, of sort of genomics as a scientific superpower, but also so that we can respond rapidly in future pandemics. So thank you again. Thanks for joining us for this session. And for those of you joining online, there's more to follow in the chat related to this session, and also follow us on Twitter during the day about this session and about the others that will follow. Thank you very much.